focus on some characteristics of pluripotency and self-renewal. Although the pluripotency genes are present in all cells, they are turned off in normal adult cells. Because it is not yet known how to turn them on, scientists decided to introduce extra copies of the pluripotency genes into normal adult skin cells in culture by infecting the cells with special laboratory viruses that had been engineered to contain the pluripotency genes. Once infected, after a few weeks, some of the cells reverted to pluripotent cells with characteristics of embryonic stem cell lines. Such laboratory infected cells cannot be used for therapeutic purposes, but these discoveries may pave the way to others to generate pluripotent stem cells from normal adult cells of every patient in need.
I kind of want to correct the statement that I made earlier. So with programming factor, I said proteins, but in this video it says genes. I mean, it's kind of technically similar to each other because you get the protein out of genes anyway, out of the transcription and everything, those kind of procedures. So what you need is eventually what the gene does, and what gene does is actually mediated by the proteins anyway. So it's pretty similar, but if you have to find the actual contributors to that induced pluripotency, is you can just say that, oh, it's genes. Anyway, so since we talked about different types of stem cells and even somatic cells, I kind of want to talk about what other factors that you have to consider when it comes to tissue engineering, which is tissue scaffold. This is very closely related to what you learn in, I guess, Monday, biomaterials, because what's tissue scaffold, the reason why we need tissue scaffold is that we want our cells to settle in our body, and the reason, the scaffold, we want to mimic the bioscaffold to mimic the naturally occurring scaffold, which is called extracellular matrix, and that ECM has many different roles that plays important things in our body. As you can tell in the slides, it has generally 8 roles, which is cell anchorage, meaning that it helps to cell to settle down and just anchor in a specific location other than just cells moving around in your body, right? And the second one is cell orientation, meaning for example, your bone cells in your legs, it doesn't grow horizontally like this, significantly, right? It grows instead vertically. So it means that cells have its own orientation when it is growing and replicating. And third one, cell growth, yes. So just like what I said earlier, cell growth grows, and the ECM is the one who actually gives the nutrients and helps the cells to grow and attach to each other and stuff like that. And the fourth one is mechanical integrity, meaning your cell, the ECM kind of helps cells to maintain its mechanical shapes and mechanical strength. Without the scaffold or ECM, the cells would not maintain the shape of it. And the fifth one is tissue microenvironment. So as you see in this picture on the right, there are a lot of things that surrounds the cells. So there are a lot of proteins, for example, glycoproteins, or even fibers that connected cells together. And this ECM is the one who creates that microenvironment for cells to survive and maintain its conditions. And the sixth one is cell differentiation. So sometimes some scientists, when they are using stem cells in the tissue engineering research, they sometimes choose specific scaffold, because the stem cell, sometimes when you want to obtain the specific cells from the stem cells, that stem cells require specific environment, and that's enabled by the specific scaffold. So that's something that you have to consider. And blueprint for tissue organizations, that's basically the same as earlier. And the eighth one is kind of interesting because you've never known that cells are communicating each other. I mean, they're not really talk like verbally or anything, but they have this like type of like method to talk to each other. For example, let's say that this specific part of your tissue requires more ATP, for example, and the source of ATP is like located over there. So like you want the other hormones or other transport protein to transfer that type of ATP over here, and that's enabled by the scaffold, which is also known as ECM, if it is natural, a scaffold. So because of those reasons, you need like really good scaffold that plays this type of similar roles in your research and in your therapeutic session. So notice that these pictures are actual, like the image of ECM, and notice that it looks very, it has a lot of big porosity, right? That's also part of the thing that you want to consider when you're designing scaffold, because if it has good porosity in your scaffold, it means that the cells are gaining nutrients and oxygen sufficiently. So, I mean, I am going to go ahead and actually like talk about the specific factor that you want to consider, excuse me, later. But yeah, the bottom line here is that ECM actually matters a lot. So the next one is, so how do you create your own scaffold? There are many different technologies out there, but I want to kind of introduce you this specific one, which is called decellularization technique. So as you can tell from its name, you're getting rid of the cells from somewhere, right? That place is ECM, of course, because ECM is what we already have in our body, and if you want to create the scaffold that is close to the natural ECM as much as possible, you want to take that original ECM in the first place, right? So what this technique does is that you're getting rid of the kind of defective, damaged cells out of the actual ECM that you already have, and the result is kind of empty ECM with that is lack of cells, right? And then attach like fresh, new, normal, functioning cells in that ECM, so that the organs, for example, in this case, in this picture, which is heart, the heart ECM is now incorporated with the fresh, new cells, and then the defective heart is now functioning pretty well later on if you recellularize it later on with the fresh cells, right? So anyway, so bottom line here is that you are not creating artificial scaffold with this technique. You're actually just reusing the frame of it and use the other cell source, for example, stem cells or somatic cells, and yeah, now you get the fresh heart. And this is the kind of time-lapse video that shows the procedures. You see that the cells are going away, and now you see the frame of it. It was possible by using the actual detergent. So you're flowing detergent in this case it was rat. I don't know if it's part of rat or the entire rat or whatever. You're flowing the detergent in that rat's body, and then now the detergent is detaching the cells out of it, and now you're seeing just ECM. And in this case, it's not the rat, it's actually human heart. And yeah, let's take a look.
For more than a decade, biologists have been able to turn stem cells into eating heart cells and petri dish. But bioengineers had some luck getting stem cells to grow on scaffolds in simple, hollow organs like tracheas and bladders. But just how hard would it be technique to make a large, complex organ with a heart from scratch. Getting beaten clumps of cells organized into a three-dimensional heart requires scaffolding. At the Massachusetts General Hospital in Boston, Harold Otten's team started with the scaffold that nature provides. They're using a technique called decellularization to strip away all the living cells from the heart. The owner of the specimen died only a few days ago. It will sit in a specialized vessel for about a week, while chemicals wash away its DNA, lipids, soluble proteins, sugars, and all other cellular material.
Building, if you want to renovate it, repair it, you go in and you have to have a drywall or get rid of cabinets and stuff like that. And basically, we're going to need to make sure something natural, but using solutions and detergents, we're getting rid of all the cells.
Afterwards, all that's left is a pale, white matrix of collagen and other structural proteins that once held everything together. Stripping away the donor cells removes the antigens that can cause the body's immune system to reject the new organ. The scaffold can then be repopulated with healthy cells that are immunologically matched to the patient. The team hopes to one day completely reconstitute working parts from the scaffold like this. But today, they're focused on how they might develop parts of the heart using the same process. They've taken a valve and a cardiac aorta from the decellularized heart and sewn it onto a cadaveric heart.
This part here is the aortic root, including the aortic valve and the ascending aorta. And this has been decellularized. So this is a decellularized graft sewn onto a cadaveric human heart. You can see the right coronary artery button and the left coronary artery button sewn onto that decellularized graft. The same thing. Here's the pulmonary artery and the right ventricle tract. Here, the cadaveric heart. This is the graft, the decellularized graft, and it's sewn onto the heart.
Harold and his team want to check if their graft will be able to do what these structures do in real life, prevent fluids from flowing back into the heart, and feed oxygenated blood from the aorta to the arteries that feed hungry heart muscles. They use a CT scanner at the hospital next door to take a look at their work. Harold pumps contrast fluid through the aorta to check for backflow and to see if the arteries that feed the heart muscle weak. The team knew an emergency department case could come through at any time, so they had to act quickly. Back in the control room, Harold runs through his handiwork with radiologist Brian Rosatra.
Here is a CT of this kind of heart, and you can very nice and injected the aortic root. You can see that the sewn storage to this area. This organism really quickly can put it with 3D mode here, and then lean it away so we can see what's left as you slow that up. So you look at the scientists together with that. He's walking to you. It looks pretty close to what I was expecting. It is like, you can see it's very nice and smooth. So what I think the most likely is the inner luminal smoothness, and it does look pretty good.
The process of decellularization and stem cell seeding could ultimately develop better replacement parts of the heart. These parts might prove more able to repair themselves and can theoretically grow with the young patient. To Harold, this technique has transformed medicine.
Last night, we were treating a patient who came in and developed some onset heart failure and is not currently on a machine that's supports her heart and lungs. So, seeing these patients and seeing that clinical need and these patients' families and understanding where all this research funds into ultimately, which is helping patients and developing tools to improve life just with these patients that have organ failure. That's really what gets us going. I think it was what keeps us going.
That was really interesting. And also, I want you to guys know that the reason why decellularization method is superior, I guess, to any other scaffoldings is that you're taking your own ECM sometimes, and if that is the case, you're avoiding the unwanted host responses, like immune responses, because that material is your own bio organs in the first place. So that your body is not going to recognize that ECM or scaffolding as a foreign material. So that's a good thing. So when it comes to scaffold, there are some design factors. Excuse me. There's some design factors that you want to consider, and the first one is cell density, meaning that if the scaffold has been seated with enough cells or not. And if there's too much of cell density in there, it means that the nutrients are not going to be delivered to each individual cell sufficiently. So if the cells are not dense enough, then the scaffold seated with the cells are not going to be functioning as much as you expected it would be. So you want to consider that cell density. And the second one is mechanical support. Is very self-explanatory. You want to choose the scaffold that is, has high strength, so that it doesn't collapse or it doesn't get damaged or anything. And the third one is transport of nutrients and oxygen or waste. You want the nutrients or oxygens to be delivered to your individual cells, considering the distance between the vessels that is applying the nutrients and oxygen to the cells, and the distance between that and the cells. So that is enabled by having some porosity in your scaffold, so that the vessels can go through the pores and deliver each individual cells the whatever they need. Actually, that porosity and the mechanical support is kind of like trade-off, because like if you have too much of porosity, then it will lose some mechanical integrity, right? So you want to kind of like consider these two factors together. And this graph is showing you the relationship between the distance between the vessel and cell and the nutrient concentration within the cells. So notice that to a certain point, to a certain distance, the concentration within the cell maintains a certain concentration, meaning that the cells are going to survive it's going to survive without dying fast. However, if it reached a certain point, which is here in 500 micrometer, if the distance is bigger than that, the concentration within one cell is exponentially decreasing, right? Meaning that the cell is not getting enough nutrients or oxygen. So meaning that the cell will die faster. So that's the relationship that you want to consider when you're designing a scaffold. And the parameter that kind of tells you the balance is Thiele's modulus, which is also written as S here. So notice that if S is bigger than 1, it means that the cell consumes more nutrient or oxygen than what is delivered to them. And if S is smaller than 1, it means that the supply is greater than the demand, so you don't really have to worry about it. And if the S equals exactly 1, then that's the balance that you want to set as a limit when you're designing the scaffold. And the next one I want to talk about is, before I mention bio 3D printing, there is a technique called cell fiber weaving before the cell bioprinting became popular. It's basically the same thing, except that it's not printing the entire structure, but you're weaving the fiber that encapsulated the cells instead. So the type of biomaterial that this technique uses hydrogels, I'm sure that you guys learned it in the biomaterial class. Hydrogel has certain mechanical integrity, but not quite. But the good part of it is that it has some good porosity inside it, that it delivers like waters or nutrients to the cells pretty easily, other than compared to for example, metallic materials. So you're encapsulating the cells within the hydrogel fiber. Of course, it should be hollow inside, and when you're doing that, when you're encapsulating the cells, you're using the microfluidic device. And after you are done with encapsulating, you want to go ahead and, of course, go through the maturation process so that the inside of the hydrogel is full of cells. And then later on, I'm not really sure if cell weaving techniques is weaving at this stage or at this stage, but anyway. So, and then you're go ahead and weaving these fibers that has the cells inside, so that you can create your, I guess, desired structure. So let's take a look at the video. It's literally weaving the fiber. So I can think of one example that this could be very useful. Like, for example, if you have a burnt skin somewhere and you want your skin to be covered faster, you can use this type of fabrics laden with cells and maybe therapeutic drugs inside, and then kind of attach that patch on your, I guess, burnt skin, and it recovers faster. But anyway, this was kind of cutting edge technology before bioprinting was available. And I'm sure you guys all know what 3D printing is, but I'm not sure if you guys ever heard of or seen 3D bioprinting, so this is the video that shows.
In just a few years, 3D printing has gone from a space-age concept to a technology you can use at home. Today I was in Philadelphia to meet with a company that has a 3D printer that doesn't print plastic toys and stuff. It prints human flesh. Welcome to Biobox. They've designed and built what could be the first step to making a synthetic human. A machine that 3D prints human tissue. And its box. So what is Biobox?
Biobox builds tools that empower people to design living things. We're doing it with the goal of eliminating human waiting lists, curing disease, remediating climate change, and even pushing back for the past.
So 3D printing for human tissue?
Exactly, yeah. That's really the first. Our first product is a 3D bioprinter, and it prints three-dimensional living tissues out of human cells.
Well, Danny and his Biobox team certainly don't lack a mission. They designed and built their 3D printers in-house and have developed a revolutionary printing process that uses living cells as the ink. The cells are mixed with a biological material that acts as a kind of glue, and after printing, the end result is a man-made human flesh.
So step number one is acquiring human cells. How are those acquired?
They're actually from the foreskin of a patient. Yes. So it's tissue that normally be thrown away, but they can isolate cells from them, and grow them up, expand them, and that's what a lot of people use.
We don't actually do any of that work ourselves, and we use the cells when we test our materials. But it is what we're doing is building platforms so that we're going to use different cell types, different cells from different sources to build more useful and different tissue.
Today we're recording here, and here you can see sort of the 3D file of what the computer would look like in the access diagram. Show you it's actually flat and will lay on the petri dish nicely.
Usually the printing would take place in a sterile environment so that the human cells would get spoiled. But as this year was going to be used, it didn't matter.
And who would be this?
We have a lot of research institutions that are using this astronauts or other types of tissue engineering. We hope that it helps get across different companies or even your local hospital eventually when it
I actually met the employee from BioBot in KC a few months ago, and he was like telling me that they are looking for software engineers or whoever is able to write a code specifically for 3D printing, because what they're using now is the software that is designed specifically for 3D printing, not bioprinting. That's what he said. So like, if you can come up with like specific protocols or software that you can use that for biobots, then the company's going to hire you right away. That's actually what he says. So that's pretty awesome. Anyway, so when you're doing 3D printing, there are certain steps that you want to take. First one is imaging. Like, of course, you want to kind of image, like what structure you want to print out, right? Either using X-ray, CT, or MRI. And then after that, you want to go ahead and kind of choose the design approach based on that image that you just did. The first approach could be biomimicry, meaning, for example, in this case, it looks like ear. So what it does is that you don't want to like print out something that doesn't look like ear and then call it ear, you know. So you want to like print out as similar structure as possible that mimics the actual biostructure. The second method is self-assembly, meaning that you're not printing out scaffold and cells at the same time, but instead just cell only, and expecting the cells to assemble itself. So that's, I guess, another approach that you can take. And the third one is mini tissue. So what it does is that it is a very, the minimum unit that function as if, what I'm saying is like it's a minimum unit that kind of function that you expect it to function. Like for example, if you want to print out renal epithelial tissue, for example, and you don't want to like print out the entire kidney, right? If you want to just test like, for example, certain drug reaction or stuff like that. So you just go ahead and like print out a small like minimum unit like that kind of has the compact functionality of the renal epithelial tissue. But that's that. Like you're just printing out the mini version of the entire system. And the third one is you're selecting the materials. Either synthetic polymers or natural polymers or ECM, as I mentioned earlier. ECM is actually the best one, but you have only a limited amount of ECM you can get because, you cannot it's naturally occurring, so you have only one structure available from one patient. And the fourth one is cell selection. That's when the knowledge of like stem cells and somatic cells comes in, and either you decide if it is stem cells, you want to differentiate it and stuff like that. Yeah, and the fifth one is bioprinting, and here's you also have to decide which bioprinting method you want to choose, like either inkjet or micro extrusion, laser assisted. They have its own advantage and disadvantage, so you want to consider that. And the last one is, of course, application. Either you're going to use that structure for therapeutic purposes or just research purposes. It's whatever. You decided. And then finally, I can kind of introduce you what type of 3D printing method you can choose. So the inkjet bioprinter, you're literally you're using either thermal heating method to discharge the materials that you have or cells that you have in the tip of the printer by using the actual heat or piezoelectric method, which means that notice that it has a piezoelectric actuator here. So you're using electrical like pulses and mechanical like pressure like converted from one another, and you're using that pressure to release the biomaterials or cells. And another category is micro extrusion bioprinter. So you're using either pneumatic or mechanical, meaning pistol or screw, to release that cells and scaffold. The last one is laser assisted bioprinter. So you're using just laser, and there is a observing substrate, that's what it is called, and that's how you generate the pressure, and that drops the cells from the plate, I guess. And yeah, so as you can see, the tissue engineering is very complicated, very interdisciplinary field of study. And also when you're printing out the cells, you have to consider surface resolution. Like you want to make sure that it has a good resolution. And cell viability, that's important, because if you print out some cells and it dies out like really quickly, then you don't want to do that. And yeah, of course, biological materials and stuff like that. So yeah, that's it. I guess it's kind of early. Okay. Any questions?
All right, well, thank you.